Well, I think it has been a very interesting symposium. We have heard very new information from different models. And right now, what Alexander just presented, I think it's a very interesting story on new regulatory transcriptions and genes which are involved in a very interesting question about aging and all this part of the life cycle of a yeast. And so we have seen along the whole session different models, and now we have arrived to a very interesting model in a plant model. And Marta Rendón also comes from LANGEBIO, just as well as Alexander. And well, Marta, she graduated from genomic sciences, and we have two representatives in this symposium, Alejandra Medina and now Marta Rendón, which both come from genomic sciences, which is a relatively new undergraduate studies, which has been very fruitful in generating different young researchers which are now already working in science and doing very fine and nice contributions. So then she made her master's degree in biochemical sciences at the Biotechnology Institute in UNAM under Dr. Posani's guidance, and then she finished making her PhD in plant biotechnology at LANGEBIO, guided by Alfredo Herrera. She is now making a postdoc in Alfredo Herrera lab, and she has made a really nice work in being which she's going to talk about, but I think as well as other of the young researchers in LANGEBIO, like Alexander, they are doing really a very nice work, and it is very interesting to hear them. Also, I mean, today I want to stress the fact that all the speakers are very young researchers which are already giving rise to new concepts, new genes, new ways of seeing things, and I think this is very nice and very important. And with that, as in Mexico, I can see how science has evolved, how the young people are really having a chance, and they are profiting from this opportunity and making very, very nice contributions. So now, Marta, please, thank you very much for accepting our invitation to be with us here and to take the part of the plants. We could not miss at least a talk on plant biology.
Plants, yes.
She says, plants are never considered. They have been considered, and particularly with your talk, which I'm sure will be most interesting. Thank you very much. Mm-hmm.
Thank you, Alicia, for this very nice introduction, and thank you, Jimena, for the invitation to attend this symposium. It's always a pleasure for me to come to the university after all these years working at CINVESTAV. So I'm going to talk about the most recent results we have produced at Alfredo Herrera's lab. We have been trying to understand common bean evolution. And I'm going to talk I'm going to discuss several aspects on population genomics and comparative genomics, which are the main tools that we have been using to understand this very, very interesting model. So, okay, so common bean is a legume, a very important legume that was domesticated several thousand years ago. And if we think of domestication in general, when we think of crop and animal domestication, we have to say that this is probably the most important human event. It started around 13,000 years ago around the world. And we have several examples of crop domestication around all along the different continents. For instance, in Asia, we have the examples of rice domestication, soybean domestication. In Africa, we have iconic examples such as coffee bean plants. In America, we have maize, we have chili pepper, we have squash, and we have, of course, common bean plants. Now, domestication is very important because it allowed the transition from gathering civilizations to agriculture-based civilizations, and this is a major transition for human societies. Now, domestication impacted not only in nutritional terms, you know, the human habits, but also it has an impact on several other aspects of life, such as in this example here, the religious aspect. This is just these are two pictures that I took recently at the Anthropology Museum here in Mexico City, showing two different deities that the Aztecs had devoted to domesticated maize plants. And what I think is really interesting from these pictures is that Aztecs had two deities, one devoted to young maize cobs, this is the one here, and another one to mature maize plants, which means that they really observed carefully these domesticated plants, and they really had an impact in several aspects of this Aztec culture. Unfortunately for me, I was not able to find any deity for common bean, which is unfortunate, but I'm sure that if we look in closer details, we might find something more, if not with the Aztecs, maybe with the Mayan culture or something else. Now, what are we talking about when we say domestication as a biological process? So, when we think of domesticated plants, we're talking about plants that have a set of traits, morphological traits, that allow us to distinguish them from their wild relatives. All these traits are known as the domestication syndrome, and it's precisely a syndrome because these traits are shared by many domesticated plants everywhere. So here I'm showing you some of the most evident examples of morphological traits that we have that emerged during the domestication of common bean. This is, for example, the loss of seed dispersal. Probably you have all seen dry common bean plants. So when we have very, very dry pods, the lignin fibers that are contained here in these pods, when they get dry, they allow the pod to curl and explode the seeds and disperse them easily. With the domesticated plants, this doesn't occur anymore. Of course, this is an undesirable trait when we think of harvesting. So we have, for the domesticated common bean plants, we have pods that do not open spontaneously, and so they require the human activities to get the seeds dispersed. Another thing is that the growth habit of these common bean plants change. Usually, I'm sure you have all seen common bean plants growing in milpas, and they use maize plants as scaffolds because they have climbing growth habits. Whereas domesticated plants like BAD 93, which is a Mesoamerican accession, change this growth habit, and now they grow as bushes. Another important aspect is that the seeds change a lot. We have very large seeds when we think of domesticated plants. This is the example of BAD 93 seeds, which are very large, very rich in protein and starch content, whereas the wild seeds are usually smaller, and they're really toxic for consumption. And another consequence of domestication and of recent improvement, genetic improvement, is also the resistance to different types of stress. So we have modern varieties which tend to have more resistant genes and allow them to grow in several conditions. For example, they can resist drought tolerance, or they're less prone to bacterial or viral infections, okay? So why is it so important to talk about common bean? I mean, it's a domesticated legume, it is a very nice model, we have very little information, but where's the importance of this plant? Well, first of all, common bean is the most important grain legume for human nourishment around the world. In countries like Mexico, it is actually the main source of protein, if not the only, for many thousands of people here in the country. Another important aspect is that this legume is very recalcitrant. It has been recalcitrant to genetic transformation, which means that common bean improvement relies on traditional breeding systems, which can take years. So the goal of farmers and breeders and molecular biologists is to accelerate these breeding programs in order to design new varieties that can, face new challenges such as climate change, the increase in temperature, we have less water, we have a lot of bacterial and viral infections, etc. So the goal here to accelerate these breeding programs is first to identify the genes behind the emergence of these domestication morphological traits, which are the alleles and the polymorphisms that allow these plants to adapt to so many different environmental conditions. And once we have all this information, all this molecular information, then we can tell the breeders, okay, these are the markers that you should be using for your breeding programs, and eventually accelerate this, the creation of new varieties. So this is where our project starts, and that's the goal that we had when we started working and doing genomics on common beans. So the first step, in order to identify these genes, which for many other domesticated plants are already known, for maize, for instance, or rice, there are several examples and well-proven domestication genes. For beans, we don't have such thing. We have no clue which domestication genes are there. So in order to identify these genes and these loci, or interesting alleles and polymorphisms behind these important morphoagronomic traits, so we had to give a first step, and that was the generation of a reference genome. Okay? And so we started working on that a few years ago, probably 5 or 6 years ago. So there was an international consortium led by Alfredo Herrera in LANGEBIO, which involved 4 different countries, Mexico, Spain, Brazil, and Argentina. And we worked all together to generate this first reference genome of common bean. So we chose BAD93, which is a Mesoamerican variety, which is very interesting because of all the resistant genes that it has, and also because it has traditionally been used as a mapping population to, for the identification of QTLs and other interesting alleles. And so here is the morphology of this accession. So we have a plant that grows as a bush. We have indecent pods. We have white flowers, which are less prone to outcrossing, to receive natural pollinators such as bees or hummingbirds. We have very large seeds. They're usually white, and so this was the chosen genotype. So last year we published this reference genome. It was a very nice and complete paper, I think. So here I'm showing you just some of the basic statistics that one has to show when talking about a reference genome. So the estimated size of the genome is around 600 megabases. We assembled around 550 megabases of this estimated genome size. We predicted around 30,000 protein-coding genes, but most importantly, we also identified non-coding RNAs, small and long non-coding RNAs, which are here. And interestingly, our assembly was complete enough to anchor 80% of our scaffolds into 11 linkage group. Phaseolus vulgaris is a diploid plant with 11 pairs of chromosomes. Not only we deciphered the genome sequence of this Mesoamerican genotype, but also we constructed a transcriptional atlas of BAD93, which means that we sequence RNA from all tissues, organs, and all along the development of the plant, right from the very early vegetative stage till the till the last reproductive stage. So, which is something that you can see here in this image, and. The analysis of these RNA samples allowed us to accurately identify which are the genes that are regulated all along the development of the plant. So here what I'm showing is just a summary of all the genes that are up or down regulated during the different developmental stages, not only the protein coding genes, but also the long non-coding RNAs, which are regulated during the development of the plant. So this information is available for all of those who are interested in working on this type of analysis. And so now we have the reference genome of this non-model legume of common bean. So what's the next question? So how do we start asking this reference genome, which are the interesting genes that we should be looking for and that we should try to fix into our new varieties that should be resistant to all these challenges, environmental challenges? So once we had this reference genome, we started to model the evolution of this plant and to ask some interesting questions that one has to answer in order to understand really, the history of the plant, and also to adapt all these natural processes such as artificial selection and hybridizations into the generation of new varieties. So if one wants to understand this evolutionary history, the first thing that we have to answer is when and where this domesticated plant, first, where it originated and where it got domesticated. This is the first question. The next thing is, how common was the hybridization between the domesticated plants and the wild relatives growing around it, and also how well did these domesticated varieties adapt to anthropogenic environments. So these are the three main axes that we have been working on. So I'm going to start talking about the very first question regarding the origin, the centers of origin and the centers of domestication of Phaseolus vulgaris. So, Phaseolus vulgaris is, as I said before, it's a legume. It belongs to the genus Phaseolus. Phaseolus has more than 70 described species so far. And there were some theories about the origin of Phaseolus vulgaris as a species, I mean. So, some evidence published back in the 90s suggested that Phaseolus vulgaris originated here in this area of Peru and Ecuador, and from here it migrated to Mesoamerica and the Andean regions in South America, where it was independently domesticated 10,000 years ago. And this observation was based on the analysis of one single gene, the gene encoding phaseolin, which is a seed storage protein, and based on the repeat content of this gene, the authors of this paper, led by Paul Gebs, said that the isoform of phaseolin encoded containing this population here of Peru and Ecuador had the ancestral isoform of this protein. Therefore, this was the origin of Phaseolus vulgaris as a species. Now, having more molecular evidence, more recently in 2012, Roberto Papa in Italy published this work where he said that actually the origin of Phaseolus vulgaris is not Peru and Ecuador, but it's Mexico, it's Mesoamerica. And what he did was he sequenced 5 different loci, nuclear markers, from several wild accessions from Mesoamerica and from the Andean region, including the Peruvian Ecuadorian area. So what he did, he concatenated these nuclear markers and he constructed this phylogenetic tree. And what he observed was that both the Andean subpopulation, which is here highlighted in red, and the Peruvian Ecuadorian subpopulation highlighted in yellow, were derived from different Mesoamerican wild subpopulations that are here depicted in different colors. So based on this observation, he said, you know what, Peru and Ecuador cannot be the origin, it has to be Mexico, and from here these populations derived. And later, of course, these were domesticated 2 times independently, one in Mesoamerica and the second time in the Andes. Now authors, research groups around the world tend to agree in one thing. There were 2 independent domestication events for common bean, one occurring here in Mexico and the other one here at the north of Argentina and the south of Ecuador. And of course, if we think of the distribution of these Phaseolus species in the continent, it's very easy to think how, yes, Phaseolus vulgaris did originate in Mesoamerica. So in this map, I'm just showing you the richness of species that we have in the continent. So what we can see here is that Mexico is the only country with these red spots here, which means that we have the highest richness of Phaseolus species. Okay, so the radiation of this genus, of the Phaseolus genus, occurred in Mexico. And later, some of these species invaded the southern hemisphere. Some of them were even introduced by humans, like in the case of Brazil. These populations are not wild populations, they are all domesticated plants that were introduced recently. Okay, so based on this radiation in Mesoamerica, it's easy to think that Phaseolus vulgaris indeed originate in Mexico. But then what is happening with these Peruvian Ecuadorian accessions that for so many years have been treated as the ancestral form or an intermediate form of common bean? So the first thing we did in order to answer this question about the origin of common bean as a species and what was going on with these accessions from Peru and Ecuador, well, we had the reference BAT93 genome, and then we sequenced 30 additional genomes, several of them from Phaseolus vulgaris, some others from more distant phylogenetic species, like Phaseolus coccineus, Dumosus, costarricensis, and some others which are less related, like Phaseolus hintoni, etc. These are all wild species that grow only in Mexico, okay? And of course we included accessions from Peru and Ecuador, which was the target population that we wanted to know if it was really, you know, the ancestral form or intermediate form, or if it was Phaseolus vulgaris at all, okay? So we sequenced these genomes, we mapped them against our reference genome, our BAT93 reference genome, And I'm not going to give you any of the technical details of this, but we obtain single nucleotide polymorphisms for this, for each of the 11 linkage groups. And we reconstructed this phylogenetic tree using around 500,000 nuclear SNPs here, using the maximum likelihood methodology. And so what we observed here was that, well, indeed, all the Phaseolus vulgaris accessions were grouped in one monophyletic clade. These species here, the rest of the genomes are grouped accordingly to previously reported phylogenies using ribosomal sequences. But what happened with the Peruvian Ecuadorian accessions? Well, they formed an independent clade, a sister clade to Phaseolus vulgaris. They were not at all derived from any wild subpopulation from Mesoamerica. This clade here contains wild and domesticated accessions both from Mesoamerica and the Andean region. So, from the nuclear markers, we could say, no, definitely these accessions are not derived from any, at least not from any of the populations that we sampled. We confirmed this observation using chloroplastic markers. So we had 1/3 of the chloroplast genome here, which it's represented by 55 kbs of the chloroplast genome. We again reconstructed the phylogeny using this information, and we have exactly the same pattern. We have here the monophyletic clade of Phaseolus vulgaris, we have the Peruvian Ecuadorian clade here, and we have the other sister species as previously reported. So based on this information, we thought, okay, this has to be some sort of a speciation event or a lineage divergence that predates the split of the Mesoamerican and Andean the split of the Mesoamerican and Andean gene pools. Now, we needed a little more validation of this observation in order to claim that we were having a sister species. So we calculated the absolute genetic divergence of these subpopulations. We grouped our accessions into different subpopulations according to the places where they were collected. And so we calculated this index, the DXY index. And so this is the genetic divergence that we obtained when we compared populations from the same species, from Phaseolus vulgaris. So this is the average genetic divergence that one could expect when comparing interspecies populations, okay? Now, when we compare Phaseolus vulgaris and how divergent it is from other sister species such as Phaseolus coccineus, Dumosus, or costarricensis, then of course we have a larger DXY index. We compare the Peruvian Ecuadorian accessions among them as one population, and we observe that it is a very restricted population, not only in ecological terms, but also in terms of the diversity that it has. Okay? And so what happens when we compare Phaseolus vulgaris and this Peruvian Ecuadorian subpopulation? Well, we have a higher DXY index, similar to the one that we obtained when we compared two sister species, clearly defined in terms of taxonomic units. And so this genetic divergence between the Peruvian Ecuadorian accessions and Phaseolus vulgaris is as high as one could expect between two sister species like these two. So again, this was telling us that this is not Phaseolus vulgaris, this is a different Phaseolus species. And so, taking advantage of these polymorphisms that we had, we also needed a temporal frame for this lineage divergence. So we used the information from the chloroplast markers, and we ran some coalescence simulations, and we dated actually the timing of the split of these lineages. So, as previously suggested by other groups from the US, the Mesoamerican and Andean lineages of Phaseolus vulgaris represented here by BAD93, a reference genome, and Jallo, which is an Andean variety. These two gene pools diverged around 160,000 years ago, but the divergence between Phaseolus vulgaris and these Peruvian Ecuadorian accessions occurred around 1 million years ago, okay? So, based on all this information, the genetic divergence, the phylogenetic signal, and everything, so we thought, okay, we can confidently say that this is another species, is the closest sister species to Phaseolus vulgaris, and now we call it Phaseolus pseudo-vulgaris. Okay, and from now on, if you see Phaseolus pseudo-vulgaris, this is what we're talking about. We're talking about the accessions from Peru and Ecuador. Now, I'm not a biologist, and I think that the definition of a new species is really, really complex, and usually we face these issues when we're dealing with cryptic species. And we're saying this is a cryptic species because in morphological terms, it is really hard to distinguish Phaseolus pseudo-vulgaris from Phaseolus vulgaris, but still we needed phenotypic differentiation of these two species. So instead of I mean, of course, we checked the flowers and the leaves and everything, but there was no trait that could distinguish these two species. So we used a more sensible method, and we ran some metabolomic profiles. So this is something that we did with Robert Winkler at CINVESTAV. And so basically we obtained more than 1,000 different metabolites from young trifoliate leaves from Phaseolus vulgaris, Phaseolus pseudovulgaris, and an outgroup which is Phaseolus coccineus. Here is what you see basically is the heat map of the abundance of these, of only 30 significant metabolites here, and based on the abundance of these metabolites, we did some clustering of these accessions, and basically this clustering reconstructed our phylogenetic results. So in one clade here we have all the Phaseolus vulgaris accessions. In an independent clade we have all the Phaseolus pseudovulgaris accessions, and here we have the outgroup which is Phaseolus coccineus. Now interestingly, several of these metabolites that differentiate between the species are flavonoids, and some of you might know flavonoids are very important signaling molecules to establish the symbiotic relationships between rhizobia and legumes. So what this is suggesting is that that changes in symbiont preferences have also accompanied the diversification of Phaseolus species, along the genus, along the evolution of this the genus. So now we have a clear answer to this origin question. So we're sure that the origin of Phaseolus vulgaris, it is Mesoamerica, that the Peruvian Ecuadorian accessions are not vulgaris, it's a sister species, which in evolutionary terms is very interesting because now we do have the closest species to what could be the ancestral form of vulgaris and pseudovulgaris, okay? So the next question we needed to answer was, what has been the effect of hybridization? So you can imagine that a species is domesticated, it originates somewhere, then it gets domesticated, and from these domestication centers, then it starts, dispersing. And so how can these domesticates adapt to different environments? Well, basically, hybridizations, outcrossing, can help a lot to adapt these new varieties when they're introduced in other places. So, which is the effect of these hybridization events between domesticated and local wild accessions? So basically, when we think about hybridization, we can have two different scenarios, which are very interesting, both of them. So from one side, we can have a domesticated population arising through artificial selection. We have the wild relatives here, and once we start growing these domesticated plants in the neighborhood of these wild relatives, then they can outcross if reproductive barriers have not been fully established. And what happens is that we have weedy populations, intermediate populations between these wild and domesticated plants. Usually farmers tend to maintain these seeds from the weedy populations and plant them again and grow them, and so the hybridization events occur every year. And the most extreme scenario when we think about hybridization through time is that new species can arise as a result of this hybridization event. So this is the case of Phaseolus dumosus, which is a closely related species, and it is a result of hybridization between Phaseolus vulgaris and Phaseolus coccineus. It's a stable hybrid, and it's now recognized as an independent species. Of course, again, this occurs when reproductive barriers have not fully established between two species, which is also contradictory because most people tend to define a species based on the reproductive isolation, but in this case, this is not occurring, not 100%, okay? So we wanted to know which is the extent of gene flow between different vulgaris subpopulations, and also what's happening with the gene flow outside Phaseolus vulgaris. So there are several tools that have been recently developed to estimate the gene flow between two species. Probably some of you read the papers on Neanderthal and Homo sapiens hybridizations. So several methods were developed to estimate the amount of genomic transfer between these two species. These methods have been applied to other species like plants or butterflies, even polar bears, producing very interesting results. So we use these methodologies to estimate the amount of gene flow and what we call genomic introgression between vulgaris and other closely related species. So here I'm showing you the FD estimator of introgression. It's an index based actually on shared polymorphisms. And what we see here, when we compared Phaseolus vulgaris subpopulations, we have a very high index, a very high FD index, suggesting that there's a lot of transfer of genomic blocks between these populations. This occurs when we compare wild and wild subpopulations, wild towards domesticated subpopulations and domesticated towards wild subpopulations. Now, this introgression signal decays as we move backwards in the phylogeny when we compare vulgaris against more distant species. In this case, we're comparing it to the Phaseolus pseudo-vulgaris subpopulation here and with Phaseolus dumosus and Phaseolus coccineus. So we can see, of course, this is expected. These species are not that closely related anymore, but still there's introgression occurring naturally. I have to say this is a picture that we're taking of this gene flow effect. So this has been a continuous phenomenon occurring all along the evolution of Phaseolus species, okay? Now, we observed several interesting things. So for example, we observed an unbalanced interspecies gene flow. I don't know if the resolution, if the colors are okay for you, but you might see some green dots here in these plots. So what we observed, each dot represents a 5 kb window of in each of the 11 linkage groups of these Phaseolus vulgaris subpopulations. And in green, I'm just highlighting those 5 kb windows which have significant introgression values. So if we compare, when we analyze the directionality of the flow, we see that there's a lower density of green dots here when we're comparing the flow from wild subpopulations into domesticated subpopulations. But we see more green dots when we're analyzing the flow from a domesticated subpopulation into a wild subpopulation, which means that there's more escape of genomic blocks from domesticated into wilds than the other way around. This can also be observed in terms of the length of the windows that are transferred. You might not be able to see here the scale, but of course the genomic tracts are larger here in this case, and of course the number of protein coding genes that are transferred during these introgression events is larger. The functionality of these genes, I'm going to talk about that a little bit later. We also observed an unbalanced introgression across hemispheres, but in this case we were comparing the gene flow. We were studying the gene flow between Phaseolus vulgaris from Mesoamerica and the Andes and this population of Peru and Ecuador, that here instead of marking it as pseudo-vulgaris, it has the tag AH. AH comes from the name of the zone here in the Andean region where it's coming from. We collected these accessions here at the Amotape-Huancabamba deflection, so that's the tag AH from Amotape-Huancabamba. And so we wanted to know which was the frequency of gene flow between these Mesoamerican and Andean gene pools towards Phaseolus pseudo-vulgaris. And again, what we observed here was, if you can see the green dots, they're not that common when we compare the pseudo-vulgaris and the Mesoamerican subpopulation. The MA is domesticated Mesoamerican, WMA is wild Mesoamerican. But what happens when we compare the flow from the Phaseolus pseudo-vulgaris subpopulation towards the Andean subpopulation? Then we have a huge density of green dots here. What this is telling us is that even though we don't have fully established reproductive barriers, the gene flow from here to Mesoamerica or from Mesoamerica to the Peruvian Ecuadorian area is highly restricted. But from here to here is much more common, which means that Phaseolus pseudo-vulgaris can be a good pollen donor, but cannot be a good pollen receptor. Okay, so this is very interesting if we want to understand the establishment of reproductive barriers in legumes, which is another subject that is a very dark thing. Nobody knows which are the factors defining reproductive isolation being transferred into Phaseolus vulgaris coming from these sister species, which is really interesting in terms of how we can use all these alleles coming from other species to take advantage of these alleles for, in modern breeding programs, okay? So based on what I just said, actually hybridization has occurred all the time within a particular species and between sister species, and this is also very interesting because Phaseolus vulgaris was supposed to be an autogamous plant, and our results are showing that yes, it's preferentially autogamous, but it can outcross and with a very high frequency with its neighbors and with other species. And the last question that we wanted to answer was how well did domesticates adapt to anthropogenic environments? So for this we had to define domestication genes basically. So what happens when we're thinking about the domestication process in terms of population genomics? So what happens basically is that during domestication there's a strong artificial selection applied on these plants, on these target plants. And so what happens is that genetic diversity is strongly decreased, and also well, actually, as a result, as a consequence of successive bottlenecks, which is what I'm showing you here. So let's say this is a wild population, and each color represents a different allele in this wild population. So as we move forward during the domestication process, you see that the colors of these boxes, we don't have the green anymore, we have less colors here, and the more modern varieties which have suffered this domestication and artificial selection for so many hundreds of years, they have even less colors than at the very beginning, which means that we have lost several alleles, we have lost a lot of polymorphisms, and so this is something that we can trace in terms of the presence or absence of certain polymorphisms or haplotypes, and this is exactly what we did. We took advantage of the fact that we're losing genetic diversity and that we would expect only a few alleles to be fixed in these domesticated populations, and that's how we defined domestication gene candidates. So basically what we did was to look for haplotype association. We looked for haplotypes, blindly, using all the genomes that we sequenced, and we checked where these haplotypes were falling, if they were affecting protein-coding genes or long non-coding RNAs, and if these haplotypes were associated or not with the domesticated phenotype. So using this method, we identified around 1,200 protein-coding genes and 100 long non-coding RNAs with domestication-associated haplotypes. And some of the functions that are associated to these protein-coding genes are really, really interesting. We have, for example, genes from the sucrose starch biosynthetic pathway, which is of course behind the changes in the size of the seeds. We had also haplotypes affecting genes from the vernalization and photoperiod pathways, genes involved in stress response, in hormone signaling, and organ formation, for example. These are some of the examples of the genes that we identified with this method. Now, what was also very interesting was that most of these domestication gene candidates are shared with other plants, other basal plants like rosids or asterids, and only 15% of these domestication genes are legume-specific genes, which is also very important in terms, if we think of the domestication syndrome, as a syndrome, which means that all these morphological traits are shared with other plants, it makes a lot of sense that these domestication candidates are also shared with other basal plants, which means that we can achieve the same phenotype, altering the same pathways or functions, okay? And so if we compare these two events, hybridization, the effects of introgression, and artificial selection during domestication, we can have a very, very interesting conclusion here. So basically what happens is that domestication, artificial selection, has targeted protein-coding genes that are behind the emergence of these morphological traits that we can easily observe and that distinguish the wild plants from the domesticated accessions. At the same time, hybridization, right from the beginning of the early speciation events, hybridization has occurred and it has allowed plants to adapt to new environments. And basically we can say this because most of the genes that are transferred between populations are adapted genes, are stress response genes. These are only some examples of the genes that we found here. So We have genes related to biotic and abiotic stresses here, and this is just an example of a pathway, of the vernalization pathway, that is affected as a result of artificial selection. So you can see this is a very interesting evolutionary scenario, because right from the speciation we have hybridization occurring all along the history of Phaseolus vulgaris, and recently the domestication has affected genetic diversity. It has reduced, importantly, this genetic diversity, but hybridization is again reintroducing this diversity into this domesticated population. So it is, it's a very interesting game of how plants are losing and regaining this genetic diversity in order to maintain these morphoagronomic traits that are important for breeders, but at the same time these plants can adapt to new environments. So, just to finish, if we put together all these signals, we have this, this is the landscape of the whole genome, and here we have, each line represents an introgress block from different populations here of Phaseolus vulgaris, Pseudovulgaris, or other species, other sister species. Here I'm showing you the domestication gene candidates that we have. These are the protein coding genes, and these are the long non-coding RNAs, for which we don't have, we don't know which are the functions of these long non-coding RNAs. We have no clue what kind of genes they are targeting, so this is something that we have to analyze carefully. So if we put the signals together, we can see that also these domestication genes act as barrier loci for recombination, as there's no overlap between these domestication genes and the introgression regions. This is something that has been observed in other plants like maize, for example. This has already been described. So, with all this information on the origin, the gene flow, the artificial selection, and how this has shaped the genomes of the varieties we have nowadays, we came out with two very interesting models, spatiotemporal models, for a common being origin, dispersal routes, and domestication. So, the first model, what we think is that probably an ancestral form of vulgaris, it emerged here in Mesoamerica, in Mexico in particular. It started invading the southern hemisphere. That's something that plants do naturally. They move and they can, disperse easily. And so this ancestral form invaded the southern hemisphere. It reached the Peruvian Ecuadorian area of Amotape Huancabamba, where it got stocked and geographically isolated. And from here, it actually speciated. It was an allopatric speciation event. In a second migration, once Phaseolus vulgaris was established as a species here in Mesoamerica, it migrated again to the southern hemisphere, and later, thousands of years later, it was independently domesticated here and here. So the gene flow has been maintained between these two gene pools, limited with the accessions from the Amotape-Huancabamba deflection, and probably also this seed dispersion was promoted by bird migrations. This is something that we have been discussing a lot. And there's a very nice study that was published last year, which modeled the migration routes of over 100 bird species along the Americas. And what we see here is that when birds move from north to the southern hemisphere, they usually cross this Amotape-Huancabamba area here. When they're coming, in the other direction, they usually don't fly over this area here. They tend to fly through the Gulf of Mexico. So this is probably explaining also why there has not been a lot of exchange between Mesoamerican accessions and the Peruvian Ecuadorian populations. And the second model, which is not incompatible with the first I described, has to do with glacial periods during the Pleistocene. So again, we have this ancestral form emerging here in Mesoamerica. It invades the southern hemisphere together with other Phaseolus species such as Phaseolus coccineus. But actually, our coalescence simulations and the timing of the split of these lineages, it matches several glacial periods during the Pleistocene. And basically what we think that happened is that these glacial periods actually eliminated most of the genetic diversity here. These events killed most of the populations, the Phaseolus populations that lived here in the southern hemisphere. The Amotape-Huancabamba deflection could have worked as a glacial refugee. And then, again, we have this speciation event occurring here actually because of this geographical isolation, and only a few populations that did survive these glacial periods expanded later and finally were domesticated several thousand years after these glacial periods. And I mean, this speciation events is not weird when we think about the Amotape-Huancabamba deflection. Here I'm just showing you, if you can see the colors here, these are the Andean mountains. This is the deflection here, and you can see here the red dots, which means that this deflection is very rich in terms of endemic species. Okay, so it's a hotspot for speciation. So what happened basically was that Phaseolus came into that deflection, it got isolated, and it diverged from Phaseolus vulgaris. So I'm not going to go into more conclusion. I actually, with these models, I gave you most of the take-home messages. I just want to finish saying that based on our results, it is clear that, first of all, we need more sequencing efforts in order to really cover most of the diversity of Phaseolus vulgaris populations that we have in Mexico. And what is more important, I think, is that now with this type of analysis, this population genomic analysis, we can start to define target populations that are more promiscuous and that can easily transfer genomic blocks from, to other modern varieties. And so I think that understanding the diversity that we have and measuring the levels of integration and hybridization that occurs in Mesoamerica, we can really accelerate breeding programs and eventually have more resistant varieties in the very near future. So I just want to finish with this slide. There are so many people involved in this project, particularly at the very early stages of the production of the reference sequence. And so just a few names here. These are the people involved from CINVESTAV, which were strongly involved in this project. Alfredo Herrera, as I said, the leader of this consortium and my PhD advisor. Luis Delaye, Robert Winkler, who helped us a lot with the metabolomic side. These two guys were postdocs in our lab, and they helped a lot understand the biology of the plant. At CRG, at the Center for Genomic Regulation in Barcelona, we have worked a lot with Roderick Guigot, Tony Gabaldon, and Cedric Notre Dame in most of the bioinformatic analysis. And I also have to thank Alfonso Delgado here from the Institute of Biology and Paul Gebs at UC Davis. Those are the real experts on common bean. They are the geneticists and the taxonomists, and they're the guys who do know the plant. And without their help, I could say we would have never finished this evolutionary story. And this is just a very recent picture of what we call the bean team. It's a multidisciplinary team that is, hopefully it will be very productive in the next years. So we have breeders here from INIFAB, Jorge Acosta from CIAT, Steve Beebe, Paul Gebs from UC Davis, Ale Covarrubias from IBT, at Cuernavaca, Alfonso Delgado here at the Institute of Biology, and Alfredo Herrera and myself, who are doing all the genomic analysis at LANGEBIO. So that'll be it. Thank you for your attention, and I'll be happy to take any questions you might have.
Gracias, Marta. Qué padre.
Gracias.
Now, can you tell us more about this very interesting, this mechanism of biased pollination? What's known about it? I mean, have people tried to start doing it? Maybe in other systems also, like greenhouse experiments in which you can actually pinpoint this particular morphological structures, or? Yeah, actually we have no clue on that. I mean, Paul Gebs several years ago started doing these crosses between these Peruvian Ecuadorian accessions and the Mesoamerican and Andean gene pools. He started observing that the fertile, plants that were produced were, he got very variable percentages of successful F1 plants. And when we look at the morphology of the flowers, there's nothing that can actually tell us what's happening there. Alfonso Delgado did a lot of microscopy with these Peruvian Ecuadorian accessions. And yeah, I mean, he found a few structures that could be, more curly and that might
stop the bees from coming and take the pollen. I mean, but these are just speculation. And we have there's only one well-known example of incompatibility, which is basically, I can't remember the name of the loci, but that explains a bit of the incompatibility between Mesoamerican and Andean populations. It's not 100% of incompatibility, but it reduces a lot. So it's a game of alleles, a dominant allele, and etc. But that's all we know. So there's no information about the reproductive barriers. So what I think these introgression analyses are very interesting, because usually what you expect is that if you're having hybridizations and recombinations and the fixation of certain genomic blocks from one population into the other, you expect them to have neutral genes or advantageous genes, okay? So genes that would, benefit the population. Of course, you're not expecting to have deleterious genes or genes that are important to define the identity of a species being transferred to the other one. So probably the fact that we're observing these barrier loci and genes that are, blocks that are never ever transferred between populations, that could give us some clues on the reproductive barriers in genomic terms. So that's all we know. So that's why I said it's a very dark issue here for the legumes.
Interesting. Can I just run another quick question, because
Sure.
it's related. When you move to domestication genes, does part of your analysis include looking at co-occurrence or co-non-occurrence
Looking for what, sorry?
for co-occurrence of these
Co-occurrence.
selection genes, so you can start making inferences on
On
compensation
epistatic
between the
events or
genes,
codominance or
epistatic,
stuff like that?
yeah.
Not yet, no.
And do
So
people look into that? Is it possible?
I think our main issue, of course we would like to do that. The main issue is our sampling. We have a very small sampling. So yeah, what we did was, the co-occurrence only inside an haplotype. So that's all. So we didn't check, if different haplotypes were occurring at the same time in different populations. So yeah, we definitely have to do that. But also the co-occurrence in different linkage groups. That's even more interesting. Not in the same genomic block. Yes.
Very interesting talk.
Thank you.
I'm curious about one of the data that you showed, which compare the number of protein coding genes that are found in the common bean with the domesticated plants that were not the common bean.
You mean this
Yeah.
phylogeny? Yes.
Correct. So the question that I have is, if you perform a similar analysis with other domesticated plants, will you see the same distribution of domesticated genes? Or the reason for asking this is because, as you said, you are interested in making these plants more easily to domesticate by genetic means. And there are examples of plants that are already easily domesticatable. And this one is the difficult one, right?
Yes.
So perhaps if you will compare this distribution with the other ones that are easier, you will understand why this one is harder than the other ones, because probably this distribution doesn't fit the PCGs that makes plants more domesticatable. That's a wild guess, but my question is, have you done some analysis like that?
Yeah, I think I'm getting your point. So, so if we think of the distribution of these protein coding genes, if they are basal to other plants and if they are shared, I can tell you that those examples of domestication genes that have been proven in other plants like maize or rice, they are of course shared also with legumes. There are transcription factors such as shatterproof, or in the case of maize, TB1. All these genes, they are all present in common bean. The problem, for example, for shatterproof, which is a gene behind the loss of seed dispersal and the hintations of the pods, we have so many copies in common bean that the one that has been identified in rice, for example, or in Arabidopsis, does not match the copies that we have in common bean. So these are these important differences that, these genes are present in all of these plants. Domestication genes are present, from rice and maize, are present in maize, in common bean. But at this point, we have not seen any haplotype association with those isoforms or gene copies that were affected in other plants. So they're present there, but simply not containing SNPs. So, as I said, my guess is that, yeah, most of the domestication genes are shared with other plants.
But those are the genes that are shared with the common bean.
Yes.
The question will be if you have other plants that are not common to the common bean. If you compare those with the ones that are similar to that plant that is already domesticated.
Right.
If the distribution that you see here is the same or different than the other ones, that will give you some clues
on
what are those genes that are making this plant harder to domesticate.
Right. Yeah, probably that would be another approach. I don't know how many other plants are studied at this detail, so for now we have only the iconic examples, but yeah, that's like the other direction in which we could ask
Trigo.
the question.
What is the name of trigo in
Wheat.
English?
Wheat.
Wheat, for example.
Wheat is an extremely complicated system for this analysis because it is a tetraploid and it has a huge genome, and I don't even know if it's a tetraploid, but it has a huge genome, and I'm not sure if there are well-validated domestication genes for this. I mean, the cases I know of is tomato, rice, maize, soybean, maybe, and I mean, for which we have experimental validation. So, yeah, I think we need more models, and in our case, as I said, as I was telling Alex, we need a larger sample in order to discriminate between these genes. But yeah, I mean, that's an interesting approach, definitely.
Okay.
Any questions? Thank you.
Nice symposium, and then we have a very good chance to organize it around the visit of Katia, and that we thank very much all of the speakers because it was really an interesting symposium, and very new aspects came out in the different systems, and I think that maybe we should do it again sometime. Katia, when do you want to come back? Yes, and well, thank you to all the speakers, and we are in touch. We have the addresses, so we can keep in touch and see each other sometime. It was very nice to have you all here. Thank you very much.